Okay, we're going to pick up sort of where we left off on Tuesday. We are working on quality control, mapping, and variant calling for genome-wide association studies, or really this will extend to when we do transcriptomics next week as well. First step is always to know whether your reads are any good, and then to map them. And in the case of GWAS, we're interested in the variance. Next week, we'll be interested in quantifying sort of the depth of the mapping at every spot to give us an indication of gene expression. Oh, and I actually get ahead of myself. It's been a busy morning already. The quizzes last night, I got a flurry of emails. I don't know how many were. I can't distinguish in the emails how many are in here and how many are in the online section, but for all the ones that I've had time to follow up on that got numbers wrong, it was, well, if it was in the single way, just the blast of one to the other, it was almost an error, always an error. Actually, every case was an error. It quit early on one of the jobs. I don't know why. Some of them said canceled. I don't know why they got canceled. I can't really tell the exact keystrokes that end up causing some of these mistakes, but one of the things, if it ran smoothly for you and you got them all right, great, but let it be a lesson to you as well. When you run things in batch, like from a Slurm script, the fact that you don't see the errors just because you get output doesn't mean it did it right. So you should always go back and look at the log files. The .o and the .e files, always check those. Right? When you're running things in the Colab notebook, if there's an error, you see it right away, right? Because it puts it in the output. But if you run a Slurm script and you get output, you think everything went fine. But that might not be the case, okay? So the lesson is always go look at your log files after a job finishes. And especially sometimes, although I didn't see these, based on what was in the error file, I'm almost sure that if they got the email probably said failed. You know how Slurm sends you a started and a completed, and it puts it in all caps. Sometimes that one that comes a few minutes later says failed and not completed, in which case you definitely need to go look at the error file and figure out, you know, what went wrong. Okay. Any other questions about the homework? I have not looked at the scores. Those will post. We have to go in and manually look at the code that got uploaded, or write scripts to look at the code that got uploaded. So those will post after the code gets graded.
For the code for the three-way, I had to take 2 screenshots of there's a Word document uploaded. Is that okay?
Screenshots.
Of my code.
Uh-huh. Okay.
I had 2 of them. I had to put it in the Word document. I uploaded it.
That will make it trickier for us. That's okay. We'll look at it. In the future, if you're asked to upload code, copy paste into a text document and give me the code, not a picture of the code. Look at the output in the .o and the .e files. It's sometimes the error messages are really obscure, and I mean, this has been run hundreds, if not thousands of times now for this class, because that three-way blast has been, I've been doing that exercise forever. So I have seen it go wrong in lots of different ways, but they tend to be pretty rare. The most common one is that it got canceled, and I never know the reason that the job canceled. Okay, so sorry, that was a false start on the what we're doing for this for next. Okay, we didn't get very far in this section, and my plan was to do interact, and we could walk through each step in the QC and mapping and all that. But in the interest of time, I decided to just give it all to you in a Slurm script. So this was posted, but not all the steps, on Tuesday. So if you downloaded this job script on Tuesday, it didn't have everything in it that you're going to want. So you should go ahead and download that now. Move that on to Bridges, where your data lives. So hopefully everybody got the data. It's uploaded to Bridges already. You've got a directory that has the SRR files and the E. coli genome in it. I guess if I go, okay, so I have lots because it's completed the whole pipeline for me. But you should have the directory with, whoops, not trimmed yet, maybe. How many of you finished the trimmed? Because that was sort of the last thing I pointed to the page where you could go try that on your own. If you didn't, that's fine. If it's in the directory already, it won't matter. It will just overwrite those files with what I've named them in the job script. Actually, it won't overwrite them. It'll add them with the names that I gave them. So all you need to have in this directory is that genome file, and it can be zipped. And then those two read files that came with it. Okay, and then you will want to open in terminal so that you open a terminal window in that directory. And yes, I changed the color of my terminal just because I got tired of looking at white on black. That's the only reason it looks different. You can change the theme up in the corner. Talked about that a long time ago, but just in case you're wondering why it doesn't look just like yours. So, right, this is my directory. Yours only has the reads, the job bowtie trimmed rm.sh, right? That's the job script that you should upload. And then the NC 012967.1 FASTA, that's the E. coli genome file. Okay, so how do we run this? Okay, so this is your job script. Job underscore bowtie underscore trimmed underscore RM. That just run it on a regular partition node. And so I'm calling it mapped trimmed. Oh, I don't know if the uploaded version has my email there. Check and see if it has my email. It has my email, so change that to your email. When we get back to the slides, I'll tell you, I modified a couple things just to make this go faster. It does all the same steps. It just isn't going to write some of the intermediate files. And in that process, I left my email in there. So just so you know, If you're not following instructions right now and you're listening later and you didn't follow instructions because you're listening at 4x speed, I'm going to get an email with your name in it saying that your job started. This is somewhat annotated, and you'll see as we walk through the slides what's going on in all of these. The only difference here is that at the beginning there's this chunk, well, obviously there's the hardware setup, the SBatch part at the top, but then there's a section where we have to go get the fast P. I talked about that a little bit last week. This code checks to see if you've already created that bioinf environment, and if you have, whether fastp has been installed or not. If either of those are not true, it fixes, right? It will create the environment and then install fastp, and then at the end it activates that bioinf environment. You've got to have all that code so that when you send this job to the production nodes on bridges, it has the software available to then execute the commands, right? Because fastp is not pre-installed. You can't just say module load fastp. So then we'll talk about these steps as we go through them. How do we run this, though? Back to my original question. So we're in our terminal, stuck at the command prompt. Yes, SBatch, and then I just typed job tab to fill it in, and then you hit enter. It's going to overwrite all my files. I don't care. And this is super fast. This will only take a couple minutes for it to run, but we're not going to wait. I'll let you look at that after we go through the slides. Something that I want to note, we are running this on 16 cores, and this was a ChatGPT thing, optimizing. Before we used -N, 128 threads, and that was actually causing problems. Some of the software apparently couldn't keep up. Like, that was too many threads. So telling it how many cores instead of how many threads was the solution, and that was just me and ChatGPT having a conversation. Well, it was me giving the errors to ChatGPT and saying, what's going on here? Because they were not interpretable errors by me. I'm like, this should not be choking on this command. Okay, so I'm going to jump back to the slides while those run. Since we just submitted, hopefully, like 30 plus jobs, you may have to sit in the queue for a minute. So I think this is where we ended on Tuesday. And I said you could try this on your own if you want to. Anybody having trouble getting the SBatch submitted? I'm always stuck in this tension in this class of I'm really excited about the next thing that I want to get to, but then also balancing that with the frustration level of the thing we're on now is not working. We should get a start and then either a failed or a completed. I think for me on whatever we're running 16 cores, it took like 2.5 minutes or something like that. Okay, so what is it doing? And if you open the job script, you can look at them side by side if you want to. But that first section is running the fastp, right? So if we were doing this interactively, this module load anaconda, what I was talking about, making sure that that bioinf environment exists, and then here. Okay, so this is Conda is the package manager for Python. These are all Python programs. FastP is written in Python, and it's just saying create this environment called bioinf, and then go to the channel CondaForge, which is a repository for Python packages. And if it's not in CondaForge, also look in the bioconda channel, and then FastP is the name of the program that we want to install. So we'll go look in those two repositories and install it when it finds it. Then you would activate the bioinf environment. In an interactive session, and actually should start the interact session before you do bioinf. You have those out of order. I'll fix that later. And you're not going to run this interactively anyway, because it's running in the background right now. The FastP right here, this is the same thing. The thread count might be different, but that's just saying how many cores do you want to run it on. Telling you the input files, and then what do you want to name the output files. Remember, FastP is the combination of FastQC. So it's looking at those quality scores for the reads. It has default parameters that we kind of know are good for only retaining the high quality base calls, and it's doing the trimming. So it's the combination of that FastQC and Trim-O-Matic or Trim Galore. I can't remember which trimming program it incorporated, but the outputs then, the trimmed reads, are -O. And then these are optional, but just so we can see what it did, I had it output an HTML file and a JSON file. The JSON is really the data. The HTML just makes it pretty to look at. And then you would exit the interact session, download those files, and this is what you would be looking at. Okay. And your batch script will generate these. So if you look in your directory after it finishes, you'll have these. You can download them and look at them just like I'm doing, just like if you were doing it in an interact session. Take homes here for this data, right? Before filtering, said there were 7.6 million reads. Okay. And then after filtering, 7.36 million reads. Right. So it got rid of a few 100,000 reads that it said were low quality. Just so make sure we're keeping track of the forest and not just looking at the trees. Why are we doing this? Why do QC? I mean, maybe it's obvious. I'm not trying to insult your intelligence, but just want to make sure we see the whole forest here. Yeah, and so why is that really important? Like thinking long term, what are we trying to do in this whole module? We're trying to identify the variants, right? At the end of the day, we want to know what are the variants in this genome. And if you think about doing this in a clinical situation, which if you become a bioinformaticist, lots of the jobs are clinical, or at UT Southwestern, you've got a research study trying to associate variants with a certain kind of cancer, right? You don't want to be calling variants that are actually just base calling errors, okay? So we're trying to get rid of all of the low quality base calls so that we're sure we don't call, or as sure as we can be, that we're just not creating a technical artifact and saying that's a causing cancer, something like that, okay? So just big picture. Okay, so what are we going to map these to? Generally, The options are a reference genome, or you have some de novo assembly of a genome. We're going to do it to a reference genome in this case. Where would you go to get the reference genome? We've already looked at NCBI for BLAST, but that's where you would go to get your genome and the annotations for that genome as well. You could also do it yourself. Lots of the labs, well, several of the labs in the biology department are interested in non-model systems. So we do some de novo sequencing, where you are doing the sequencing and the assembly, talked about on Tuesday. I'm not going to talk about those because those are mostly one-off projects. Next week, we're going to talk about transcriptomes, so we will get into assembly a little bit. And in some cases, in non-model systems, you can assemble at least all the protein coding genes from RNA-seq data. Talk about that next week. And then you can detect the variants from that kind of pipeline as well. Or you can be building on a low-quality public reference. So this is... there are lots of genomes out there that are really bad. So you could just be adding data to a currently available genome, in which case you would sort of be iterating on the assembly before you call the variants. It gets pretty messy there. We're going to pretend we've got well-curated reference genomes. So what I mean by really messy, not very good genomes, a lot of them are fragmented. So this short read sequencing is not very good for getting whole, very large contigs. You'll see genomes that are only sequenced with Illumina that have tens of thousands of contigs, not chromosomes. Okay, even if they have genetic sort of recombination data to say these things are close to each other, you don't have contiguous sequence across that whole chromosome. So that's messy. And why that's relevant for us is even if we have 7.3 million reads, if the assembly that we're trying to map to is all chopped up, it's not very well assembled, a lot of those reads won't align because we've sequenced from the actual genome, and the sort of representation of that, the reference genome, is not contiguous. So we get one read from here, and then there's nothing assembled to that, so we can't map it. Okay, and so those kinds of bad quality reference genomes are a problem. E. coli, not a problem, which is the example data that you have. Many bacteria, less of a problem. As we get new technologies that have longer reads, I mentioned PacBio on Tuesday briefly. This is a long read technology. The assemblies are much, much better. Okay? So, 10 years, this may not be a problem, but it's still a problem, especially if you're in a non-model system. And then the other thing about if you're in a non-model system, there might not be an annotation. And if there is, it might not have all the genes because, when I first started working on the beetles, there wasn't a genome. And then even after we got a genome, the annotations for the genes were really bad. When I talked about how part of the reason that I probably got this job was my gene family evolution work, that was back in the early days of the chimp genome. We had a terrible time getting that stuff published because everybody said, the chimp genome is so bad, you can't infer the orthologs between human and chimp, right? Everybody knew that the chimp genome was not well annotated. And so, right, that's just the problem of if you're at the cutting edge in a given system, the genome is brand new, you're going to run into these kind of headaches. Even if you can map variants because it's well assembled, you don't know if that variant is in a gene or not. Practically, so now we're moving out of the quality control and into the mapping part. How do you map? Well, you need your reference genome, then you need a mapping algorithm. BLAST is too slow for the scale of NGS, right? We have, even in this E. coli sort of toy data set, we've got 7.5 million reads. Think of that as having to do 7.5 million BLAST jobs, okay? You've got to take each of those reads and blast it against the genome to see where does it come from. That's pretty slow. So lots of cool computer science has gone into how do we make that fast. Mostly comes down to indexing. You know, how do you compress the data into as simple a structure as possible? The two main ones that get used for aligning Illumina sequence to a reference genome are Bowtie2, that's the one we're going to use today, and then variants on BWA. So there's BWA SW, BWA MEM, BWA MEM2 is the most recent one. There are a couple papers saying that you should use BWA MEM2 on eukaryotic genomes when you can, because it deals with indels better than Bowtie2. I'm just like, if you're doing this for a real project, read the literature. What's the latest on what's the best aligner or the best mapper? For small bacterial genomes, Bowtie2 is a little faster and not appreciably different in its calls. I've run it both ways for our data. It's the same, so we're going with the faster one for now. So Bowtie2 is what we're going to use. There's almost always a trade-off between the accuracy and the speed. So Bowtie2 a little faster. That means I would assume that it might not be quite as good. But for bacteria, they're all pretty much... I say this because I'm used to working on eukaryotes, which everything takes an order of magnitude longer because the genomes are so much bigger and more complicated. In my life, the bacterial genomes are all easy. And so it doesn't matter as long as you're using BWA or Bowtie. For especially long read stuff, Cactus is a new one. I put this in here just because this is the latest sort of widely used mapper. And it's specifically for whole genome alignments, not necessarily for short read. How does it work? It uses the same strategy as BLAST. The first step is, you know how the first step in blasting was the make blast DB, where it basically breaks down the database into all of it tokenizes it. In the same sense that we talked about way back when we were doing large language models. It breaks it down into small words and creates a distribution of those words. So this is going to do the same thing where your reference genome is indexed into those words and your reads are indexed like that. And so then it's just matching distributions as a start. So it does the same kind of go look for the best match in some short word and then extend it. And because the reads are pretty short, and the tuning, because we expect them to be basically exact matches, it can go fast. I guess the analogy here is, if you remember back to our simple pattern matching where we had, you knew the small pattern and it had to match exactly, that was way faster than when you started allowing for mismatches, right? So there's a lot of computation in the allowing for mismatch and how closely does it have to match. This is essentially doing a quick search for everything that's close by in terms of the word space, and then is it an exact match? Yes or no, move on, okay? what that means is there's a difference between the mapping score and an alignment score. So something can map well, meaning this is your most likely map position, and it still doesn't align well necessarily. Or, yeah, for short reads, that's almost always how it goes. Paired end mapping. There's a lot of information that you can get from paired ends that you don't get from single ends. Because you get information about how well did the pairs map together on a genome. So, for example, you'll get things that have the wrong orientation or the wrong distance apart. And that's all information about whether there have been insertions and deletions or rearrangements in the genome. And so most aligners will give you a bin of discordant pairs. So these are pairs of reads that fall outside the expected distribution, given the read orientation we talked about. Same thing with singletons and orphans. That's like when only one of the reads in a pair can map. All good for structural variant detection. Okay, so if we were now going back to doing this in Interact, I would say go ahead and run Bowtie. You'd need those files. And if you look back at your job script, you'll see a line that looks like this, sort of. One of the speed up sort of efficiency things that I've done in the job script is to not write out. I've tied the bow tie to the SAM tools. So this doesn't end with writing out this summary or the SAM file. All this is telling it to do is, right, how many parallel jobs are we going to run, read 1 and read 2, and then what to output in terms of the SAM file, and send the summary to a file instead of to the screen, which is what that last bit does with the greater than sign. The 2 greater than sign is saying write the SAM out, the summary to that file. And you would get something like this. It says 96.92% of the reads aligned. And you can see how many of them were paired. Basically just all the statistics you might want to know for how well did your data map to the reference. And so, why is that relevant? You might sometimes, and maybe we'll come across this next week in transcriptomes, where you get only like 80% of the reads map, or less than that. That should sort of raise a red flag to you about there's something wrong. Like my data did not all come from what I thought it did. Why only 80% of the reads that I just resequenced this person and only 80% of the reads mapped back? Any guesses as to what the number one source of that problem is? I've got 7.6 million reads, and what if only 80% of them map? What's going on with those other 20%? There are 2 main things that you should suspect, like right off the bat. One is contamination, right? That is, I've got bacteria in my, like when I was doing library prep, somebody was messy, somebody sneezed in the tube, and so I've got DNA from something else. Original beetle genome had cow DNA in it because they were being sequenced at the same time at Baylor. And who knows how that cow DNA got in the Tribolium library, NGS library, but it did. And so we had a big config in the genome that blasted, and it had like a no, it was higher than that. It was like 85% match to cow. So contamination is usually my first guess. I'll take all those reads and blast them to see what they hit. There are also softwares to screen to run it against a bacterial database. The other one is that you're not sequencing exactly the same thing. So you're sequencing from a different population or a different strain, and there's enough genetic variation between what you're resequencing and the reference genome that those reads are not mapping. There's just too many variants in there. Okay, so it doesn't recognize where it's supposed to come from, and it just doesn't map. So anytime you see a low percent mapping, you should start to question what's going on with your data. If you don't do the quality control, okay, always do the quality control. But if you didn't, you might also see lower mapping rate, right? Because you've got a bunch of errors in the data. Those are going to prevent some of the reads from mapping. Just a little bit, we're not actually going to look at the SAM file in this job script. I didn't have it save the SAM files. They tend to be really big. But this is an example. If we were going to use less to look at them, you'd see millions of lines of that. They're in something called cigar format. This is a super condensed nomenclature that gives you information about what the read was, what the quality of the read was. It's pulling those straight from the FASTQ. And then where does it map in the genome, and whether it maps with the strand, and does it have gaps to interpret that. This is one of those where ChatGPT is so nice, because it can just tell you, it understands all of that code, not in the sense of programming code, but in the sense of the abbreviations. Right. So this string right here, 2S, 5M, 2I, that is the condensed format of how this alignment matches to the reference genome. Right, 2 substitutions, then 5 matches, then 2 insertions, then 4 matches, and so on. Okay, right. So that's the mapping. There are a couple steps to convert that into a BAM file. So you'll see the SAM tools section of the code. So I skipped over that. Definition slide. When you see SNV, sometimes you see SNV and sometimes you see SNP. SNP, single nucleotide polymorphism. SNV, single nucleotide variant. So using variant is a little bit more broad, because you don't know whether the variant, like polymorphism, means something about how it's segregating in the population. Also, just because it says single nucleotide variant, it really just means it's small. Like a 2 base pair indel still shows up in the SNV file. What it really means is it's a variant small enough for most reads to be able to span that variant. So small indels get binned in with SNVs as well. Okay, so next thing, we're going from that output of bowtie. So we've got our mapped reads. Now we're going to call variants. This is the converted into a BAM part. A lot of this is taken out of the job script because we didn't write intermediate files. So these SAM tool steps, if you go look at your job script, they just get piped. So you get bowtie piped into SAM tools, So SAMTools view, SAMTools sort, and then you index it at the end so that we can use that BAM file, which is the same as SAM, it's just machine readable to make it fast. Like if you do less on the BAM file, you just get gibberish, a bunch of unrecognizable characters. Last couple steps, BCF tools is the package that does the variant calling. So this is just giving you the characteristics here. In the job script, I've switched it to the more updated, the -M consensus calling. The details between those two don't really matter to us. So what is mpileup doing? This is how variants get called. So it piles up all the reads that map to a specific location and says, what's the probability of there being a variant there? You'll see reference and alternative alleles output in the variant call file. Hopefully it's intuitive. Reference genome, the reference allele is the one that's coming from the reference genome. If there's an alternate allele, right, that's the one in the alternate column. The reason I point that out is when you get into the human genetics literature, especially, you'll see minor allele frequency. So the MAF, that's the allele that is less than 50%, okay? It's not always the alternative allele, so you just have to kind of be careful of that. So the question here is that A that the red line is pointing to a variant? Is that C a variant? What do you think? Would you want to call that, let's just say this A right here, should we call this a variant? And you might be saying, well, yeah, of course we should. There's an A there, and the reference genome has a G. What are the problems with just doing that? How about that? If this, let's pretend this came from a single individual, what's the expectation if this individual actually had a G and an A? So this is a heterozygous individual. What's the expectation for the read frequency? If you have one copy of A in every cell and one copy of G in every cell, and you randomly draw to make the reads, what's the expected frequency? 50-50. Yeah. Okay. So the fact that I got 1 out of 12, this is the question for variant calling. This is why it's not as simple as just saying, well, are there any things that don't match the reference? Do we want to call that? Do we want to call this individual a heterozygote? Only 1 read out of 12 supports this being heterozygous. So the variant callers look and say, what's the distribution? And you tell it, what's the ploidy of the individual? It depends. Like if it's a haploid, right? So we're doing E. coli. Is there any chance that this is a heterozygote? No. Okay. So that A is probably not a true variant. Okay. It's just a base calling error. And, why, how do you get a base calling error? Well, there are a lot of steps between taking those pictures, making that library, and where we are now in our pipeline. Okay, so that's the problem with variant calling, is where do you draw the line? How do you determine whether this is a true variant or not? Okay. So the BCF tools is doing that. The more sophisticated ones will take the regional base calling quality, the individual base calling quality as factors, and whether it calls it true or not. It gets, if you do pooled sampling, right? So you got more than one individual, that makes it more problematic. But some of them will take those kinds of Hardy-Weinberg distribution probabilities into it. We have called variance. Questions? Staying with me? So, did your jobs complete? Do you have the data? Okay. So, you should be able to go in. Did it generate the HTML? Yeah, so you have this file. So that is the pipeline from resequencing data to calling variants. You know, if you're going to do this as part of your final projects, this is, or, in your labs for your research, other than minor tweaks to which software package you're going to use, like you may use BWA MEM, um, there are some other variant call softwares besides the BCF tools ones. But the pipeline is essentially the same. It's just kind of plug and play. All right, let's look at. We've got 7 minutes left. I'm going to introduce the GWAS stuff. So on Canvas on today, if I can find Canvas, which notebook? Right. So on Thursday in Module 7, there's a PowerPoint just called GWAS PPT. And then I've given you a link to 2 Colab notebooks. One is just called GWAS Tutorial. That's the one we're going to run. You don't need PopStruct right away. So GWAS is the one that does all the work. Yeah, so this is the GWAS PowerPoint. What do we do with our variants? Back to the very beginning, the idea here, the biological question is there a genetic basis for some phenotype? Okay, so if you want to look at what has been done, at least and especially in the human genetics world, there's a catalog of genome monitors, and this is a cool, just pure computer science project in terms of the website. Why won't? Right, but it's like you can't see it because it's so small, but each of those little dots is mapped to a certain location, and you can click on that dot and it will take you to that GWAS study. There have been tens, if not hundreds of thousands of GWAS studies now in human. And Jada, like if you're interested in a particular gene and you want to know if anything's mapped to that gene in terms of a GWAS study, somebody's found an association, this is where to go. Just to see what has already been done. The basic workflow, and I'm going to talk about all of this in the context of human. So if you're in a non-model system, you'll just have to do your best to abstract and extrapolate in your head. Hopefully some of you will do this. You'll have applicable data and we can do this and see some of it for final projects. Pipeline is the same, okay? Just different phenotypes. And if you decide you want to do a quantitative phenotype, this is a binary phenotype. You're a case or you're a control. You have the disease or the trait or you don't, okay? So that only matters for the statistics of the association. We're going to do logistic regression because it's a binary trait. But the first step would be to find your cases and find your controls. We'll come back to why that's not as trivial as just find people with the disease and people without it. Okay, there's some other factors that you might want to consider. Going to get the DNA, whether that's blood, tissues, cells, whatever, and then do the genotyping. So the genotyping is what we've done up to 5 minutes ago, right? We got our DNA, we did our QC, our mapping, and our variant calling. That is the genotyping. At the end of that, you get a matrix here. Actually, you have a variant call file. So this is a conversion of that variant call file into a matrix that just has individuals. And then the RS numbers are SNP numbers or SNV numbers, just an ID number for that variant. And then the last column is whether it's a case or a control. In our example, we've got that in a separate file, and we're just going to combine those in the background computationally. But ultimately, then what you're trying to do is say, is a particular SNP, right? In this case, SNP number 4 is the one that's highlighted there, and 5 to a slightly lesser extent. See how they match between, like, if you're at SNP 4 and you have a C, you go in the case group. If you've got a T, you go in the control group. That's the association we're talking about, right? How well associated is the variance at that SNP with the bin of the phenotype you're in, whether you belong to a case or a control. So 4 and 5, they associate really well. If you scan across the whole genome, that's how you end up with the Manhattan plot. Okay, so our SNPs are located all over the genome. This, like how high up on the y-axis, that's the negative log p. Think of that as the statistic, the p-value. You take the negative log just so higher numbers are more associated, just because it's more intuitive. Right? There's no real reason why you have to do that. It's just a prettier graph in some sense. So it turns points that are really high, that's a SNP that has a really high association with that phenotype. Okay? So that's a Manhattan plot. The other really common plot you see is called a QQ plot. And what this is showing is if all the associations were random, what's the expectation of test statistics would fall on this diagonal. When you start to see that there are p-values that are too low relative to the random draw, that means there's some signal in your data. There's actually some association there that you might want to look at. Okay? And then this is just another, these are examples of real human GWAS studies where, this is the ideal. So, for example, bipolar, you see nothing really sticking up very high, but coronary artery disease, there's a SNP that jumps way above the background level, right? That is a strong candidate for a causative SNP, right? Right now, you can just say there's a strong association, okay? But it jumps way up above the background. Just because we're at, and we'll pick up here next Tuesday, but on Canvas, the links to those Colab notebooks, you should be able to just run this. And I think it's got text in there for the instructions that you need. Again, this is you playing with that. I will post the assignment. I didn't post an assignment because I didn't know how far we were going to get. I wanted to have the assignment about GWAS. I'll just have it about running a Colab notebook that gives you a visualization of your E. coli variant call file. So really, you'll just put the VCF in this Colab notebook, and it will print a pretty picture of your variants. Okay, so that is just to tell me that you have completed variant calling up to this point. Okay.